Who was the first human-Neanderthal hybrid? The honest answer is we don't know. We will almost certainly never know. There's no name, no specific fossil we can point to and say, this is it. This is the one. The first child born of a Homo sapiens parent and a Neanderthal parent lived and died tens of thousands of years ago, and their bones are likely dust. Finding that specific person would be like trying to find a single specific grain of sand on all the world's beaches. It's a statistical impossibility, but that's the boring answer. The real story isn't about a name. It's about how we discovered that this event, this first hybrid, even happened at all. And it's about what that discovery tells us about who we are. It completely changed our understanding of our own past. It turned a simple story of one species replacing another into a much messier, more interesting, and more human story of contact, mixing, and family. For a long time, the picture was simple. Our species, Homo sapiens, evolved in Africa, as Neanderthals evolved in Europe and Asia. We were two separate branches on the human family tree. Then, around 60,000 years ago, our ancestors began to expand out of Africa. They moved into the Middle East, then into Asia and Europe. Everywhere they went, they met the Neanderthals who already lived there. And the story went, we outcompeted them. We were smarter, better organized, had better tools, or better language. Within a few thousand years of our arrival, the Neanderthals were gone. They were a dead end. An evolutionary failure. We were the sole survivors. This was the clean, straightforward narrative. We were us, they were them, we won, they lost. There was a clear line drawn in the sand between our species. Any mixing was considered impossible, or at the very least so rare it didn't matter. They were a different species, after all. Like a lion and a tiger, they might be able to produce an offspring, a liger, but that offspring is usually sterile. A dead end. Most scientists assumed the same would be true for us and Neanderthals. The first cracks in this story came from the bones themselves. Paleoanthropologists would occasionally find fossils that didn't quite fit. They looked mostly like an early modern human, but with a few strange, robust features that seemed more Neanderthal-like. Or a late Neanderthal skeleton with a slightly more delicate build than expected. A famous example is the skeleton of a child found in Portugal at a site called Lagar Velho. The child lived about 24,000 years ago, thousands of years after the Neanderthals are thought to have vanished from the area. The body was clearly that of a modern human, but the skeleton, especially the legs and jaw, was incredibly robust. The proportions were short and stocky, much more like a Neanderthal child than a sapiens child. The scientists who found it argued this was evidence of hybridization. This child, they said, was part of a mixed population. The Neanderthal genes hadn't just disappeared, they were being passed down, mixed into the local human gene pool. It caused a huge debate. Others argued that it was just a particularly stocky human child. Kids vary, they said. You can't base such a huge conclusion on one skeleton. The problem with bones is that they are open to interpretation. Two experts can look at the same jawbone and see two completely different stories. The evidence was suggestive, but it wasn't proof. To really know, you couldn't just look at the container. You had to read the recipe inside. You needed the DNA. For decades, getting DNA from ancient bones was science fiction. And DNA is a fragile molecule. When an organism dies, its DNA starts to break down immediately. It gets chopped up by enzymes, eaten by bacteria, and damaged by water and heat. After a few thousand years, you're left with nothing but tiny shattered fragments. And even worse, those fragments are buried among billions of DNA fragments from all the bacteria and fungi that have lived on the bone for millennia. It's like trying to reassemble a single-shredded book from a library that has been flooded and then composted. And on top of all that, there's the problem of contamination. Every scientist who touches the bone, every speck of dust in the lab, is covered in modern human DNA. Contamination can easily fool you into thinking you're reading an ancient genome when you're actually just sequencing the DNA of the guy who excavated the bone. But in the late 1990s and 2000s, things began to change. A team led by a Swedish geneticist named Svante Pääbo pioneered incredible new methods. They worked in ultra-clean labs, like those used for making computer chips. They wore full-body suits to avoid contamination. They developed techniques to find and isolate the tiny, broken scraps of ancient DNA and to tell them apart from the bacterial DNA and the modern human DNA. It was a slow, painstaking process. Their first big success was sequencing the Neanderthal mitochondrial genome. The mitochondrial DNA is separate from the main genome in the cell's nucleus, and there are many more copies of it, which makes it easier to find. The results from that first study seem to confirm the old story. Neanderthal mitochondrial DNA looked very different from ours. It suggested our two lineages had split a very long time ago, maybe 500,000 years in the past. There was no sign of mixing. The us versus them story seemed to hold up. But the real prize was the nuclear genome, the 3 billion letters of DNA in the cell nucleus that make up the main blueprint of an organism. It was a monumentally harder task. Pabo's team needed more bone, and better preserved bone. They got it from Vindija Cave in Croatia. They used tiny drills like a dentist to bore into the bone and extract powder from the inside where it was less likely to be contaminated. They used new, powerful sequencing machines that could read millions of DNA fragments at once. And they used sophisticated computer programs to piece the puzzle together, comparing the fragments to the already sequenced human and chimpanzee genomes to figure out where they belonged. In 2010, they published the first draft of the Neanderthal genome. It was a landmark achievement in science. For the first time, we could read the genetic recipe of an extinct human, and they did exactly what you would expect them to do next. They compared it to the genomes of people living today. This is where the bomb dropped. They compared the Neanderthal genome to the genomes of people from different parts of the world. They looked at a person of French descent, a person of Han Chinese descent, a person from Papua New Guinea, and 2 individuals from Africa. The logic was simple. Since Homo sapiens evolved in Africa, and Neanderthals lived in Europe and Asia, if there was any interbreeding, you'd expect to find traces of it in the people whose ancestors left Africa. You wouldn't expect to find it in the people whose ancestors stayed in Africa. And that is exactly what they found. The genomes of the French person, the Chinese person, and the New Guinean person all contained chunks of DNA that matched the Neanderthal genome. But the genomes of the 2 Africans did not. The amount was small, somewhere between 1% and 4% of their total DNA. But it was unmistakably there. The conclusion was inescapable. Our ancestors, after they left Africa but before they spread out across the rest of the world, met Neanderthals and had children with them. Those children, who were the first human-Neanderthal hybrids, then became part of the migrating human population. And as that population expanded into Europe, Asia, Australia, and the Americas, they carried those Neanderthal genes with them. That's why a person in Paris and a person in Beijing and a person in Peru all carry a similar amount of Neanderthal DNA. They all descend from that same founding population that mixed with Neanderthals somewhere along the way. Suddenly, the story was rewritten. It wasn't us versus them. It was us and them, together. The Neanderthals didn't just vanish. A part of them survived. It survived in us. The billions of people walking the earth today carry a living genetic memory of these ancient encounters. This discovery opened up a new set of questions. If it happened, where and when did it happen? The genetic data gives us a clue. The fact that almost all non-Africans have Neanderthal DNA, and in roughly the same amount, points to a single main period of interbreeding. The most likely location is the Middle East. It's the geographic gateway out of Africa. As modern humans migrated north into the Levant, modern-day Israel, Syria, Lebanon, they would have encountered Neanderthal populations that had lived there for a very long time. This was probably the main stage for these first encounters, somewhere between 50,000 and 60,000 years ago. So, we can start to paint a picture of who the first hybrid might have been. They were likely born in the Middle East around 55,000 years ago. One of their parents was a new arrival, part of a small band of Homo sapiens pushing out of Africa. The other parent was a native, a Neanderthal, whose ancestors had lived in those lands for 200,000 years. What would that have looked like? We can only guess. Was it a violent encounter? Or was it a peaceful one? Maybe it was just a practical arrangement between 2 small groups of hunter-gatherers, sharing resources, forming an alliance. Maybe a man from one group and a woman from another formed a bond. Or maybe it was just a chance meeting, a brief affair. We can't know the social context. But we know the biological result. A child was born. A child with a mixed heritage, the first of many. This child would have grown up in a world of Ice Age hunters. They would have learned to nab stone tools, to track animals, to read the seasons. They might have looked slightly different from the other children in their group. Perhaps a bit stockier, with a broader nose or a more prominent brow ridge. But they were, crucially, accepted. They grew up and they had children of their own. And they passed their unique mix of genes on to the next generation. That is the key. For the Neanderthal DNA to have spread so widely, these hybrids couldn't have been outcasts. They must have been successfully integrated into human society. For years, this was a story told only by the statistics in our modern DNA. We could see the echoes of these ancient events, but we didn't have any of the individuals involved. Then, in 2015, the science took another leap. The team at the Max Planck Institute in Germany analyzed a jawbone found in a cave in Romania called Pestera cu Oase, the cave with bones. The jawbone, known as Oase 1, was about 40,000 years old. It looked mostly human, but it had some Neanderthal-like traits. When they sequenced its DNA, they got a shock. This individual had a massive amount of Neanderthal DNA. Not the 1 to 4% we see in people today, but somewhere between 6% and 9%. The way the DNA was distributed was even more telling. The Neanderthal segments in our genomes today are tiny and broken up. That's because every generation, when DNA is passed down, it gets shuffled and recombined. Over thousands of generations, long ancestral segments get chopped into smaller and smaller pieces. But in the Oase1 jawbone, the Neanderthal segments were huge. They were long, unbroken chunks. The geneticists could use the size of these chunks like a stopwatch. They could calculate how many generations had passed since the Neanderthal ancestor had lived. The result was stunning. The Oase1 individual had a Neanderthal ancestor just 4 to 6 generations back. That's a great-great-great-grandparent. This wasn't a distant echo. This was immediate family history. This person, walking around Romania 40,000 years ago, was the direct recent descendant of a human-Neanderthal pairing. This finding was incredible. It made everything real. It wasn't an abstract event 55,000 years ago in the Middle East anymore. It was happening in Europe, and much more recently than previously thought. It suggested that the mixing wasn't a one-time event. It happened multiple times in different places whenever the two populations met. The Oase1 individual is the closest we've ever come to finding one of these early hybrids. They weren't the first, but they were a direct product of the process. Interestingly, this person's lineage doesn't seem to have survived. They don't appear to be a direct ancestor of modern-day Europeans. They were part of a wave of humans that moved into Europe, mixed with the local Neanderthals, and then later died out, replaced by other waves of human migration. The story gets even more complex. In a cave called Denisova in Siberia, scientists found a tiny fragment of a finger bone and a couple of molars. They didn't look like much. They could have come from a Neanderthal or a human. But when they sequenced the DNA, it was something else entirely. It was a new, previously unknown type of ancient human. They were named the Denisovans, after the cave where they were found. And of course, the scientists compared their genome to ours. They found that people in East Asia, and especially Melanesia people from places like Papua New Guinea and Australia, carried Denisovan DNA in addition to their Neanderthal DNA. Another meeting, another story of mixing. Then, from that same cave came the most remarkable discovery of all. A small, unassuming fragment of bone about 90,000 years old. They sequenced its genome. The results were so clear, so unbelievable, that the scientists thought it must be a mistake. They did it again. The results were the same. The DNA showed that this individual's mother was a Neanderthal, and her father was a Denisovan. They found a first-generation hybrid, a direct child of two different kinds of ancient humans. They nicknamed her Denny. She is the single most powerful piece of evidence we have that interbreeding between different human groups wasn't a strange, rare event. It was common enough for us to find the bones of a direct offspring against all odds. If these groups were meeting and having children, it stands to reason that Neanderthals and Homo sapiens were doing it too. Denny shows that the biological barriers weren't really there. At a genetic level, all these groups were just different flavors of human, and they were all part of a single, tangled, interbreeding population. So, what does this all mean? What did we get from these encounters besides a new understanding of our past? That small percentage of Neanderthal DNA wasn't just silent baggage. It had an effect. Some of it was probably bad. There may have been hybrid sterility, especially in males, where certain combinations of genes from the two different backgrounds didn't work well together. Scientists have noticed that there's a distinct lack of Neanderthal DNA on our X chromosome, which might be a sign of this. But some of it was good. Very good. Neanderthals had lived in Europe and Asia for hundreds of thousands of years. Their bodies were adapted to the local climate, the local food, and crucially, the local diseases. When our African ancestors arrived, they were entering a whole new world of pathogens. Interbreeding with the locals was a genetic shortcut. It was a way to pick up useful immune system genes that were already adapted to the region. And that's what we see in our DNA today. Many of the Neanderthal genes that are common in modern humans are related to our immune system, specifically genes that help us fight off viruses and bacteria. Other genes we inherited seem to relate to skin and hair. They are connected to keratin, a protein that makes up our skin, hair, and nails. Perhaps these genes helped our ancestors' skin adapt to different levels of sunlight or colder temperatures. They gave our ancestors a survival advantage in these new lands. Of course, there's a flip side. Some of the Neanderthal genes we carry are linked to things that aren't so great in our modern world. They're associated with a slightly increased risk for things like type 2 diabetes, blood clots, depression, and even allergies. These traits weren't necessarily bad 50,000 years ago. A tendency to clot blood faster would be great if you were frequently injured while hunting. An overactive immune system might be great for fighting off parasites, but in our modern environment of sterile offices and high-calorie diets, these ancient adaptations can cause problems. They are a relic of a different time, a genetic mismatch for our current lifestyle. So we can't point to a single person and say they were the first hybrid, but we can see their ghost. We see it in the genetic makeup of billions of people. We see it in the fossils from Romania. We see it in the proof of concept provided by Denny, the Neanderthal Denisovan girl from Siberia. The first human Neanderthal hybrid wasn't a monster. They weren't a freak. They were just a child, born to two different but not so different parents. They were a bridge between two worlds. That child, and others like them, played a crucial role in our story. They were the channel through which ancient genes, adapted for life outside of Africa, flowed into our species. They helped our ancestors survive in new and challenging environments. The story of human evolution is no longer a simple tree with neat branches where one branch dies off and another grows to the top. It's more like a tangled bush or a braided stream. Lineages diverge, and then they come back together. They mix. We are not a pure species that replaced everyone else. We are, in our very DNA, a hybrid. We are the product of these ancient encounters. Who was the first human Neanderthal hybrid? We don't know their name, but we know their legacy. It's written in our genomes. It's part of what makes us human. They weren't a footnote in our history, they were a part of us, and they still are. And searching for them has told us more about ourselves than we ever could have imagined. It has shown us that the lines we draw between us and them are often blurry, and sometimes they aren't there at all.